going to be as an adult. There are environmental factors. Food, as I mentioned, is probably one of the most important environmental factors. 30 to 35% of all cancers are related to diet. That's what you eat many times a day. You are exposing yourself and your genome to your environment through the things you eat. There are cultural factors. And when I say cultural factors, not just ethnicity or religion, but the culture of poverty versus the culture of affluence. There are lifestyle factors, as I mentioned, whether you're a couch potato or whether you're physically active. All of these are going to take this initial genetic component here and it's going to tweak it up, tweak it down, tweak it back up again. There'll be lots of interactions through these filters, so you'll get something coming out the other end that's going to look like you. And what this looks like will determine whether you're healthy or whether you're ill. And you can see that this doesn't happen overnight, but this happens over your lifetime. So the bottom line here is it's unlikely that any single gene, single nucleotide polymorphism, any mutation, any biomarker or risk factor will have a positive predictive value for chronic disease and certain types of cancer. I was told one time, I think Kevin brought this to me, that heart disease, one of his professors told him that many people think that heart disease is a disease of the elderly, that when you get old, you develop this cardiovascular disease. And his professor told him that actually maybe cardiovascular disease is a disease of the young, that it starts when you're young and over a long period of time and in response to negative environmental triggers that you'll end up with heart disease. So this term here, positive predictive value, is a very important term because what we're talking about is trying to identify those factors, whether they're biological factors or whether they're social cultural factors, that allow us to predict back to determine whether your children are at risk for type 2 diabetes, cardiovascular disease, asthma, certain types of cancers. That's the value of this, to look backwards, look at the youth who show no signs of these diseases and try to predict whether they are at risk. And if they are at risk, sometimes it's just a matter of tweaking the diet slightly, cutting down the amount of trans fatty acids maybe, cutting down the levels of cholesterol, upping the levels of roughage. It's not I don't believe we're going to have dramatic changes in our diet, but there'll be subtle changes. Everybody is talking about the health benefits of various types of food. So this slide shows you some of the top 10 or 11 foods that researchers are looking at because they have bioactive compounds. Most of these compounds, whether it's lycopene, catechins, resveratrol, are related to reducing two things, heart disease or cancer. A couple of these, like the omega-3 fatty acids and conjugated linolenic acids, are involved in either preventing cancer or they have a positive effect on cognitive function. They help with memory, they help with learning and that sort of thing. Of course, a soybean has been known for many years to have isoflavones, which are believed to be an antioxidant involved in reducing rates of cancer and other things. As I mentioned earlier, these are wonderful developments. We are now finding out that these wholesome, healthy foods have these additional health benefits. Some of your grandparents may have told you should eat more of these things, more broccoli. Don't steam it, stir fry it, because if you steam it, you lose all of the benefits of broccoli, the sulforaphanes. And as I mentioned earlier, that to blend this ideal, optimal diet is a little bit expensive. So we know that there's genetic variations. There are good things in foods. Some of those foods are accessible to us, some of them are not. But what's next? And what I believe is next is the individualized nutrition and genome-based dietary recommendations. That means at some point in the near future, you can take your children and you can get genotyped and somebody's going to make a recommendation that you should eat a little bit more of this, a little bit less of that. Probably you should exercise more. Make these slight adjustments in your diet so that you can avoid those chronic diseases. Now, are there any examples of that right now? And the thing is that there's a number of examples. Most of them come out of the pharmaceutical industry. It turns out that about 30 to 35% of all drugs are destroyed by your liver. You take a drug, whether it's a painkiller whether it's an antidepressant, there are enzymes, phase 1, phase 2 enzymes in your liver, which will that will degrade those drugs. So Roche Holdings has developed what what they're calling an amplichip, based on a genetic variation in one of those enzymes, CYP2D6. This is an acetylating enzyme in the liver. And they can determine whether you're going to respond well to a particular drug or whether you're going to destroy it. In the example that I've used many times of people who go to the doctor's office or the dentist's office, they get a shot of Novocaine and it has no effect because your CYP2D6 enzyme is rapidly degrading that particular drug. So it's nice to know that ahead of time, and they can give you a slightly different kind of drug. There's a lot of talk about Bildil. This is Nitromed's new drug to prevent heart failure in African Americans. It's a combo drug, drug that promotes nitric oxide and another drug that stabilizes nitric oxide. In the middle of the phase 3 trials, they found out that this drug was so effective for African Americans that they decided to stop those trials and remarket this drug for African Americans to prevent heart disease in African Americans. And it's caused a lot of controversy. People have called this the black pill, race-based medicine. But what it reflects, two things, because it's a combo drug and it's affecting two different biological processes, it reflects this complicated systems biology approach, that things are connected to each other. Secondly, it reflects a genetic variation. Now, they haven't identified that variant yet, but they eventually will. And lastly, there is a test for the use of Taxol in Japan. Taxol is probably one of the most promising anti-cancer drugs for women with breast cancer. In the United States, many women are given Taxol for breast cancer. In Japan, half the women that receive Taxol for breast cancer end up getting another disease called granulocytopenia, which is a loss of leukocytes in the blood. It's a very serious disease. So now the Japanese have a test for another CYP enzyme in the liver that will determine whether you're going to have this adverse reaction to Taxol or not. But again, isn't it nice to know this ahead of time? This goes back to what we were talking about, that positive predictive value. There are strong positive predictive values in these tests. Can we have a similar test at some point in the future for foods? So I just wanted to mention that, the concept of nutritional genomics, this link between genes and diet, is not just my idea, but many of the experts in the field have been talking about this for a number of years. One of our investigators in the center, Dr. Ron Krause, said about 4 or 5 years ago that there's increasing awareness of the potential for genetic variation in individuals to influence nutrient requirements and biological responses to nutrient intake. So what is he saying here? That we are genetically different, and that we're going to respond to our nutritional environment or our food in slightly different ways, and that can actually have effect on whether we're healthy or whether we're going to have a disease. Dr. Bruce Ames, another member of our center here, talking about single nucleotide polymorphisms. Here is an example of one. 2 identical pieces of DNA, except for this nucleotide pair right here, which is different on that chromosome. He says that single nucleotide polymorphisms provide a powerful tool for investigating the role of nutrition in human health and disease and can contribute to the definition of an optimal diet. That's interesting. What is an optimal diet? Is it going to be an optimal diet for everyone, or will it be an optimal diet for you, and you'll have a slightly different optimal diet, and we'll know that because we'll understand all those genetic variations that you bring to the table. So what does it entail to get to this stage where we're talking about genome-based dietary recommendations? First, we have to have a multidisciplinary approach to studying diet-gene interactions, and that includes the cultural, the socioeconomic, and behavioral factors, as well as all the biological factors, the technology for low-cost, high-throughput SNP analysis, sequencing, and gene expression profiling, access to databases for large, diverse human populations that include dietary and medical histories, genotype, gene expression data, and metabolic profiles on various human bodily fluids. Fourth, access to information from studies on model animal systems, bioinformatic tools and theories for dimensionality reduction and visualization of these large data sets I just described up here. A systems biology approach to investigating the relationship. And that is, remember, systems biology means that everything's connected to everything. You tweak one thing, it'll have an effect over here. And lastly, we need to use best practices for obtaining, storing, and handling confidential information involving human subjects. And that's really one of the purposes of this course. What are these best practices here? So there are a couple of statements I want to leave you with and then a couple of questions. First of all, good nutrition is a statement. Good nutrition is the cornerstone of health and disease prevention, but good nutrition is not always accessible, or as I like to say, good nutrition comes at a price. 2, dietary interventions will play an increasing role in the prevention and treatment of disease,